I'm going to talk to you a bit about the activities of machine learning at Starlab for doing brain health. Starlab is, well, this is the outline of the talk. I will give you a bit of the context of the company, and then I will give you an overview of application of machine learning in health and healthcare. And then I will give you 3 examples of how do we use at Starlab machine learning for health applications. All focus on brain health, which is our topic. So Starlab is basically a company, a technology transfer company that tries to transform scientific results into products or services that have a positive impact in society. And the main focus of the company is this one, the electrical brain. This should be a I was thinking. Bueno, it's basically the fact that the brain, the stimulus are transforming the brain into electricity, and this data in form of electricity goes from one part of the other of the brain in form of electrical impulses. So this allows us, on the one hand, to read the brain activity, and on the other hand, to apply electrical stimulation to modify and to modulate this electrical activity. I will talk a bit about this, but mostly I'm focusing on how do we measure this electrical activity with devices that we have developed in the past, and which is the kind of systems that we are applying on them. So I'm talking about basically brain health. Brain health, it's a set of diseases that affects 20% of the world population. We are talking about epilepsy. We are talking about neurodegenerative diseases or mental health, like in the case of depression. And there is a fundamental need, which is the fact that most of these diseases are based on the subjective evaluation of the symptoms. So, and this is a problem. Probably because of this fact, the development of drugs for CNS treatments, central nervous system treatments, are the riskiest business in the pharma company. So they have the lowest chance of approval at regulatory stages, and they have the longest development years. And because of that, this is the most expensive business in all the activities of the pharma companies. So we try to help here by providing biomarkers that are based on electrical activity. And we are doing this in a set of different funded projects where we developed seed concepts that we turn then into products, and I will show you some examples about that. So basically what we have is basically a brain that generates electrical activity. We record this activity with some devices that are called EEG, electroencephalography, that turn this electrical activity in the brain into digital signals. And with these digital signals, what we do after is apply machine learning algorithms to provide different services. Why is this interesting? So applying machine learning in health, I will give you some data. For instance, the 48% increase annually in health care data. So we are talking about genetic data, we are talking about neuroimaging, but also electronic health records. So this is a very interesting fact. This is maybe the reason why in the magazine Forbes was forecasting that in 2019, artificial intelligence, big data analytics, and related technologies will have a very profound impact in healthcare. Not only from the business point of view, but also from the scientific point of view. This graph is a plot of the years from 2005 to 2019 of the number of publications in PubMed, which is a repository of medical science papers, These are the numbers of publications that mention machine or deep learning, or that comment or present works on machine learning and deep learning. So there is an exponential growth. And in the top 6 specialities of this profusion of machine and deep learning algorithms, there are neurology, psychiatry, psychology. This is important to take into account. Not only this, but also the FDA-approved applications in the last years have increased. So 39 applications are approved, and some of them, exactly 18%, are applications for neurology, psychology, psychiatry, and include, for instance, diagnosis of sleep disorders, memory assessment in the elderly, or autism diagnosis. And this is also all following a trend in the field which is called biological psychiatry, which is basically trying to come from a subjective evaluation-based diagnosis to a data-driven one, which fits very well in the purposes of machine learning. So for what can we use digital markers? And this is an interesting paper in Nature Neuroscience of 2017, and offers a very clear scheme of how can you use these digital markers in the healthcare domain. So basically, you can do early detection of diseases. You can do diagnosis, differential diagnosis as well, and you can predict the treatment outcome in CNS drug development. I will show you now 3 activities that are related with these 3 different application fields of these digital markers of works and projects that we have developed at Starlab. And I will start with one which is for the early detection in Parkinson's disease. So our aim, after having developed the machine learning system, is to provide a decision support system for clinicians. So you cannot see the video, unfortunately, but basically what you have here is a subject, a patient suffering from a disease, a sleep disease that's called REM behavior disorder. If you have this disease, basically you have no inhibition of your muscle activity during the REM sleep while you are dreaming, and therefore you start moving, you start injuring your bed partner. So it's a grave disease that appears more or less when you are 60 years old. And the patients of this disease later on, in average 8 years later, develop either Parkinson's disease in 30% of the cases, or dementia with Lewy bodies, which is a particular type of dementia, or remain RBD. So the idea of applying machine learning here is, are we able to predict, by having data, EEG data, brain activity data of the people being diagnosed as RBD, are we able to prognose their neurodegenerative disease 8 years later? So that was the purpose of the grants that we applied for the Michael J. Fox Foundation in the States. And we developed a study with 7 different partners all over the world, trying to develop this integrated platform for collecting their EEG data repositories, centralizing them, and applying machine learning on this data. The machine learning system has not a lot of secrets. So, I mean, it follows the usual structure of a machine learning system. You have a stage for feature extraction. You have a stage for feature selection. Then you have a classification stage that tries to transform what's the original raw EEG data into a subject score, into a risk score for doing the diagnosis of RBD, or the prognosis of the neurodegeneration. So trying to give you a risk score that you will develop this neurodegenerative disease. And in some cases also, which one of them, Parkinson's or dementia with Lewy bodies. For analyzing EEG data, basically you follow a similar approach as you do with audio data. You split all the spectrum, which is what you have here. This is the spectra of the EEG signals in the different groups. And you split this spectra into different frequency bands, and you compute the power in these different frequency bands. And this is what you use as the features for your system. So you have 14 different channels in your EEG, and you have 13 frequency bands, so you have a feature space of 182 components. Therefore, we have developed a feature selection stage, and I will present very briefly here one of the methodologies that we have applied for feature selection, which is based on deep autoencoders. Basically, we have a network of 3 decreasing layers in dimensionality and 3 increasing layers in dimensionality, where you try to map the input and the output. And since the intermediate layer has a reduced dimensionality than the input space, what you will do is use the activity in this intermediate layer for making the input in the classification, which is in our case a support vector machine. And these are the performance measures when comparing with our wrapper or a filtering approach of the deep autoencoder. As you can see, it outperforms the other approaches, but it has a problem in operational conditions, and it's the computational cost. One interesting aspect of the system is that we are using a methodology which is called lift per subjects out, or lift per out, which was proposed in 2010, which is basically you take one example of class, the positive class, and one example of the negative class, and this is your validation test. What allows this is to give a better estimation of the performance when you have small data sets. It allows you, which is very interesting, to compute the area under the curve without having to go through the representation of the rock space. And it keeps balance in the training set because you are taking one example of each of the classes. So what you have here is the subjects of one class, the subjects of the other class, and the obtained performance in terms of area under the curve when you are testing the system. Another aspect I wanted to comment is basically the fact that there are a lot of ways of computing performance measures. If you take here, you have 3 different accuracy measures of your system, all show accuracy in your system, but basically the different figures depend on the way you are conducting the validation and on the way you are normalizing the scores. And this is often an aspect that it's not very well disclosed in the papers, in the literature. So take into account this. Basically, these are the results with a data set of 200 subjects of RBD. You can see here the performance measures. For instance, I will underscore the result of the conversion prognosis, distinguishing those patients that remain RBD from those that develop the neurodegeneration, with a performance of 82% in terms of area under the curve and 91% of right detected subjects. So in 91% of the subjects, we are able to right predict if they are going to convert into Parkinson's disease or dementia with Lewy bodies. Another interesting aspect is to use the output of the classifiers as it would be another biomarker used in clinical research. And so for this, it's interesting to give you not only the performance, but also the effect size of your system. What we are trying to do with this machine learning system is integrate it in a decision support system. All the development is very interesting from the point of view of the machine learning and from the data science point of view. But the problem is, if you don't have a neuroprotective drug, what is the sense of telling a subject that he will or she will develop a neurodegeneration in 8 years? So the business case for such a decision support system in the case of prognosis, it's very difficult. Yeah. So not everything, it's intellectual work, but also trying to find a way of making this useful. I'm going to present you very briefly our work on ADHD. As you may know, ADHD is a disease, a pediatric disease, mostly pediatric disease, although there are also adults suffering from ADHD, that has a very large misdiagnosis rates of 22%. So basically what we are trying to provide is a diagnosis for knowing, based on data, if these children or these adults are suffering from ADHD. For this, we are using a very particular type of neural network, which is called an Echostate Network. It has 3 layers, an input layer, an output one, and in the middle it has a so-called reservoir, which is a fully connected layer that randomly connects one node to the other. It's a type of recurrent neural network. And basically, the hypothesis is that we can discover chaotic synchronization between EEG channels. I will talk a bit about this. If you have 2 linear systems coupled linearly, what you will find out is synchronized systems. So this would be one variable, the other variable. The relationship is linear. Then you can apply correlational analysis, and it will deliver you a correlational coefficient. But if you have 2 chaotic systems coupled, you have another type of synchronization, which is called generalized synchronization, which delivers such a form. This will be one variable, this will be the other one. And the question in all the work was, can we use this type of recurrent neural network to characterize 2 time series that are chaotic related? This has, we have conducted a theoretical study that you can find in the journal Chaos, and we have tried to apply this concept to generate a marker for ADHD. This is our data set, 51 children, 21 with ADHD, 30 with controls. And basically what we are trying to do in the system is to, we have intervals where the children are closing their eyes, we are measuring the electrical activity, and other intervals where the children are opening their eyes. And we use the Echostate network to find out which are the intervals in which the person is opening eyes and which is the intervals in which they are closing their eyes. And we use a performance measure as a measure of the connectivity of the generalized synchronization between 2 channels in the EEG. So I'm going a bit into details because I've heard that some people were complaining about lack of content, so I'm trying to give a bit of more of this. After doing this particular connectivity metrics can be used to distinguish one group, the ADHD, from the healthy controls with a significance, with a statistical significance that you have to correct for multiple comparisons, and this has delivered another paper which hopefully will be published in Psychiatry Research. I'm going to change a bit the scope. I'm going to present you another project, a research project that we are coordinating from Starlab, which is a consciousness study. It is a PhET project, basically that has the vision that consciousness, human consciousness, will be someday measured by means of measuring the electrical activity in the brain, and also that we can alterate consciousness levels by applying electrical stimulation. This is a bit of the background on consciousness. You have here different states of consciousness displayed in a plot that is used in neurology. You have here Coma. These 2 dimensions are wakefulness and awareness. You have states of coma, so where you don't have wakefulness, you don't have any awareness, and you have conscious wakefulness here. And in between you have other states, like for instance different sleep stages here. You have lucid dreaming, which is a stage in which you have high awareness because you are aware of the content of your dreams, but you are sleeping. So that's why the index of wakefulness is here. And you have other states related with coma, minimally conscious state and vegetative state, where the subjects are awake, but they are not aware. So what we are trying to do here, and what it's related with the machine learning system, is a system related with a type of treatment that we are developing based on non-invasive brain stimulation. We apply an electrical field in the skull, in the cortex, and we are expecting this electrical field to change these minimal conscious state patients into wakeful patients. So we are conducting this study with the Coma Science group in Liège. Basically, they have applied this treatment to their patients, from which 6 out of 46 patients were responding positively to this treatment. And the question for the machine learning is, are we able to predict those patients that will respond positively from those that won't respond positively? Because if I know in advance, then I don't need to go through the therapy for these patients, which are very complex to treat. So this is another type of digital marker, the one that it's acting for predicting the treatment outcome. Basically, we are taking the EEG, the electrical activity before the treatment, and we are applying the regular machine learning system. So we are splitting again the EEG signals into different frequency bands. You have here a PCA projection of the non-responding, the red class, versus the responding class, which is quite mixed. If we use another type of approach, which is this TSNE, we can do also a projection of the feature space that delivers this other plot, which is basically you have the responding in green, the non-responding in red. And as you see, this is a problem where you have a circle in the middle, and then so more or less the other things around. And therefore, we thought we may be using support vector machines. This is a video I cannot display, but as you can remind or recall, support vector machines are good in turning nonlinear classification problems into linear ones. And that's why we apply it here. In this case, we apply leave one subject out validation. So we are taking always all the data of one subject out of the training phase, because we think this is part of the, these are reflecting the operational conditions where you are going to use your system later on. So basically, there will be a subject that with whose data has not been used for train that will be presented to the machine learning system. And the area under the curve in this case, in 100 repetitions of this cross-fold validation stage, was more or less 84% of area under the curve. Okay, and now to finish some take-home messages, just to remember for what can you use digital markers, not only of EEG activity, but also from wearables, from other types of data that is related with your health. You can use them for early detection. I've shown you how to use it in Parkinson's early detection. You can use it for differential diagnosis, as in the case of ADHD. And you can use them also for predicting treatment outcome, not only in the case of non-invasive brain stimulation, but also of drug treatments. And I would like to thank all the people of the team which have developed parts of the work that I'm presenting today. These are their names, the members of the Neuroscience Business Unit. And I would like to thank you, everybody, for your attention.
Thank you very much. Question on the EEG-related work. Can you get localized information from your EEG data to be able to differentiate what part of the brain the your signals are coming from?
Yes, you can do by applying source localization approaches, which basically project your activity in the cortical surface, or what are reading the electrodes, into other parts of the brain. But since the dimensionality of the space in your inner space of the brain is much larger than your electrode space, you need a large number of electrodes. However, we have been able to apply such approaches with 16 electrodes. So, and then with this projection into deeper areas, what you can do is correlate, for instance, we have used it in the case of the Parkinson's project. We have correlated one particular cluster in the spatial domain with the neuroimaging modalities that we have, and they are localized in the same place. So it's a kind of validation.
Thank you for interesting speech. Tell me, please, why do you use Echostate neural network in your model?
We are interested in using Echostate networks because we think that the type of relationship between the two time series of two channels in the EEG are not related linearly, as in this case, but they are related in a generalized synchronized way. So they present this type of form when you relate one to the other. So basically, if you think of EEG activity, this is coming from some dipoles that are oscillating in your brain, and they relate to each other with, or we expect to relate to each other with this type of generalized synchronization. And therefore, we think that a recurrent neural network will be able to characterize this kind of synchronization.
I noticed that in the beginning you mentioned a number of related mental disease. I also noticed...
Which kind of disease? Mental disease.
Mental disease, right? I also noticed depression, which has very large numbers, but I did not notice later on you build a model for that. So did I miss it out, or it's too complex to build out?
You are not missing anything. We are missing the data. So, so basically, for having or developing this kind of systems, you have to count with a data set that it's important, not only a data set related with depression, but in our case, also EEG data set. And this is something that we don't have so far. We are conducting now a clinical trial in the UK for treating depression with electrical stimulation of the brain, and then we expect to have this data set, this EEG data set, to develop such a system for decision support.
Another question. So what do you see as the role of brain-computer interfaces in both the treatment and, I guess, the research of the different disorders that you guys are looking into?
Yeah. Brain-computer interfaces are being used for rehabilitation of stroke. They are being used as traditionally for communication of computer with any kind of devices, wheelchairs, computers, so on. The problem of this kind of systems, first is the reliability. Second is the business case, because normally people that need this kind of brain-computer interfaces are people that are very much paralyzed, and this is our very low prevalence diseases. And therefore, for a company, it's very difficult to base the business case on a very low prevalence disease. For the rehabilitation of stroke, I think there are better possibilities, because this is a larger prevalence, and we expect to develop brain-computer interface to a larger level, and then to translate into systems also for communication for these paralyzed patients. However, we are using very much one particular modality of brain-computer interface, which is affective computing. This is something that we are using very much, and that we have a large demand on this kind of systems. These are systems that are able to measure the emotional response of a subject in front of a stimulus. It can be an advertisement, it can be an online buying process, it can be a product, it can be a fragrance. So all these are cases that we have developed in the past for clients. And this particular case of brain-computer interface, this has a very interesting applicability in the real world.
Thank you. I have a comment about Parkinson's disease. Did you think about incorporating other variables, like, for example, age, in the predictor? Because age is actually very important. I mean, there is some natural neurodegenerative processes that go on in the brain that can help you even improve those models. Is included in the predictors that you have at the moment?
Not at the moment. So what we have conducted so far is only with the EEG data. But of course, any variable that will help to increase a bit the performance would help. But the most important problems at this stage are not so much the performance, but first the regulatory process, validating your diagnosis or the decision support system, and the other problem is who is interested in the system and who will pay for this system.
Exactly.
These are the two main problems that we face at the moment in the Parkinson decision support system, more than increasing performance. That is something that we think that we can achieve by incorporating other variables or any other type of data.
I think on the validation, it is known that Parkinson's disease has about a 50% misdiagnosis rate. It's actually very difficult to diagnose the disease. There are a lot of mistakes done by the doctors with quick assessments, even in your data sets. So you're running against that problem because your gold standard already for training is not, gold standard per se. So I think your numbers are performing well for what the data that you have.
Yes, this is a circular problem. So in the sense that if you are using supervised approaches, then what you are doing is basically reproducing the diagnosis that a doctor has done, a diagnosis that includes as well a misdiagnosis rate. In the case of ADHD, 22%, in the case of Parkinson's, 50%. So to which extent are really useful these supervised approaches for developing this kind of system? So we ask ourselves, and we are trying to find also solutions to this by changing to other kind of approaches.
I think it's when biology will provide some blood-based biomarkers that you can use as gold standards. Even if there are invasive tests today, then you can train your models, and in the future we don't need to do those invasive tests anymore. We just can really rely on the digital biomarkers.
Yeah, but if you think also in the way you group the data when you analyze a biomarker, the grouping of the data is done based on the diagnosis, on the original diagnosis. Of course, if you have a misdiagnosis rate and you don't have enough sample size, this will be reproduced as well in the biomarker, no? in the physiological or in the biological one.
Chicken and egg